Hi everyone. For those people who haven't met me, my name's Courtney, and as Chris mentioned, today I'm going to be talking about straightening out reed bentgrass, Dioxia quadraceta, and talking about a new species of Dioxia from southwest Victoria. And sorry if I'm sort of motioning wildly at the screen. Online presenting is a bit difficult. So let's make a start. The first thing I'd like to do is acknowledge the traditional owners of the country that this work was completed on, and also where we did field work. So that's the Wurundjeri and Boonwurrung and the Gunditjmara people. And I'd like to just pay my respects to the elders past, present and future. So a bit of context. We're going to be talking about Dioxia, which is a grass, so it's in Poaceae. It's a Southern Hemisphere genus. It's in the subfamily Poideae, which is the biggest subfamily in Poaceae. It's in the subtribe Agrostinidae, depending on who you ask, and other genera in that Agrostis, Lachnogrostis, Polypogon, and others. And it's characterized by single-flowered spikelets. In Australia, we have 35 species of Dioxia, and 34 of these are endemic. So the only one that's not endemic is Dioxia quadraceta.
Courtney, you're on mute.
Courtney, I'm not sure if you can hear us, but you've put yourself on mute, or someone has put you on mute accidentally, I'm afraid.
Okay, I'm back.
You are indeed.
I think that might have been someone else. I'll go from the top of this slide.
Sounds good.
So yeah, quadraceta is the only species of Dioxia that's not endemic in Australia. It's got an Australasian distribution through southern Australia and New Zealand. And in New Zealand, it's quite variable as well. It's been suggested that there is probably a montane and a lowland form. And the images on the right are kind of a typical-ish looking quadraceta. So focusing in on Victoria, again, quadraceta is really variable and really widespread. It typically occurs in poorly drained areas within a really wide range of vegetation types, but it typically doesn't occur in tall forest or high alpine situations. So what we've got here is just a little screenshot of a revision of Dioxia by Joyce Vickery from 1940. And this is some of the sort of most recent published work on Dioxia, the genus. And in this revision, she described several species of Dioxia, quite a few of which are still recognized today. And she also described 8 forms of Dioxia quadraceta, and also a variant restricted to WA. So some of the characters that she used when she was describing these forms were anthesize, the degree of scabrousness of the lemma, things like panicle shape, whether it was lobed or cylindrical, and also the degree of induration of the lemma. So that's sort of the opacity and the hardness of the lemma. So Vickery's forms were never formally described, and we don't recognize them today, but they definitely say something about the huge amount of variation that we do find in quadraceta, and you can see some examples of that here in herbarium specimens from Melbourne. So you've got like the spindly form, the second one, which is maybe a more typical quadraceta, the third image, which is a really small thing, and then these really robust, like highly lobed forms. And so looking at this variation and also some stuff that's come out of New Zealand, it sort of seemed that this warranted investigation to say, is this just one species? Is all of this variation habitat related? Or do we have subspecies or something else? So using the characters that Vickery suggested, and then also characters commonly used to distinguish between Dioxia and other genera within Agrostinidae, we developed a set of characters to conduct a morphological analysis using herbarium specimens from Melbourne. So this involved a lot of measurements. So we had around 20 or 22 characters, I think, and that included things like awn length, the length of the callus hairs, the width of the culm, ligule length, anther size, and I measured around 200 herbarium specimens. We have around 400 quadricetas at the Melbourne Herbarium. And this took around 4 to 5 weeks and equated to around 4,500 measurements. So this cartoon is a fairly accurate representation of how I kind of felt at the end of each day when I would leave my microscope after like measuring, you know, hundreds of lemmas. And so using that data set, these were the results that we got. So this is an ordination. Each of the points in the ordination space represents one herbarium specimen, and the distance between the points is how similar or dissimilar they are to one another based on the set of characters that we developed. And you can see in the map the spread of samples that we used. Lots of stuff from Victoria and Tas, because that's kind of primarily what's in the Melbourne Herbarium for Quadriceta. And the other thing that you can see from the ordination is that there isn't really any distinct clusters. So this was surprising if you sort of cast your mind back to those herbarium specimens I showed and the huge amount of variation that was there. I think we kind of thought, yeah, we'll do heaps of measurements, we'll do the ordination, and we'll get a really obvious splitting out of maybe a few different subspecies. And that's obviously not what happened. And so we were kind of scratching our heads saying, you know, how can this thing and this thing be the same species? And as I've been going through each specimen and doing the measurements, I've been kind of sorting them out into groups based on a more qualitative sort of vibe, I suppose, like, oh, these are all have really fine leaves, and these are all really strongly lobed, or the point of attachment of the anther is halfway up the lemma instead of at the base of the lemma. And I was also using notes on the specimens, so people like Buglehole, Chesterfield, von Mueller, and more recently people like Dave Olbrecht, they made comments on their herbarium specimens saying, you know, slender form. And so I'd organized them into kind of groups. And there was one group of specimens that really jumped out to us. They all had this kind of fine leafed form that you can see in the second image. And they were also all from the same area. So they were from Southwest Victoria. And once we sort of started taking a closer look, we noticed that they had quite distinct seed characters. So the seed on the left is a typical Quadriceta, and the seed on the right is the seed from this slender form, which I'll just refer to as Dioxia southwest. And so So what we did was we added a new set of characters to our analysis that were mostly qualitative things to do with the seeds. So whether or not it had raised nerves on the pallia and the lemma, and if the pallia bulged out from between the lemma. And these were the results that we got. So we started feeling a bit more confident. You can see that this is only a subset of the original data because we had to go back and measure the 10 new traits or whatever it was on everything. And you can see basically all the samples for the southwest form, the blue dots, are clustered down in the corner of Victoria near Portland area. And so we started feeling pretty confident that we probably had something different. And these are just some photos that I took of the grouping that I had made, and you can see they've all got this really fine, sort of weak panicle, really skinny, so atypical of Quadricida. So we took some SEMs of the seeds, and then it sort of really became obvious. You can see here some of the characters that we measured. So the top two images are from the southwest form, and the bottom is Quadricida. And you can see these raised nerves on the pallia, and they're also running down the side of the awn. And you can see the way that the pallia bulges out from the margins of the lemma, and in contrast to Quadricida, where the pallia is kind of enveloped within the lemma, and the lemma margins kind of curve in. And you can see the callus hairs are much more prominent on Quadricida. So some of the other things that we looked at with the SEM were, we looked at the lemma surface. So again, the top two images are the southwest form, and the bottom is Quadricida. And you can see that southwest has these much more pronounced hairs on the lemma surface than in Quadricida. And the pictures on the right are of the Caryopsis, and Quadricida down the bottom has this really pronounced hilar groove along the Caryopsis, which is a lot less obvious in the southwest form. So once we were feeling fairly confident that we had something different and we knew where it was, we went out into the field to Kaboboni National Park and had a look for it. And this is the grass in the image on the left. It's not very exciting. I think it's exciting, but you know, it's not sort of very exciting to look at. And this is just a sort of blurry photo of one of the panicles, and you can see those really pronounced veins along the pallia and the lemma. So where does this new species of Quadricida occur? We collected it from Kaboboni and Lower Glenelg area. There is one collection from the Otways, I think from around the 80s. We weren't able to go there to confirm that it's still there. But it's interesting as well because the specimen from the Otways is sort of a much more robust looking thing, like it almost, the panicle is a bit more like a typical Quadricida, but then it has these seed characters of the southwest form. This is one of the habitats where we found the new species. That's Neville in the back there. It was growing in these kind of dried out, what we assume are seasonally inundated ponds or points at the end of drainage lines in what is a fairly wet landscape. And the grass was growing on these mounds at the base of what I think is Eucalyptus ovata. And some of the associated species here, aside from the ovata, we had Valaciaria reniformis, Ranunculus inundatus, and Lepidosperma longitudinale. And what was really interesting here was that it was actually co-occurring with Quadricida, and so that probably tells us maybe why it's been overlooked in the past. The other habitat that we found it in were these red gum swamps. This isn't an image from when we went there, but this is the area we found it in, and so you can see just like how much water is on the ground. So this species clearly likes these really kind of damp, seasonally inundated places, and these were actually quite heavy peaty soils, and so it's associated with these soils that have, you know, really high organic matter. And the other species that we found here, Acacia melanoxylum, Eryngium vesculosum, and Lobelia bucoliae. So yeah, it clearly really likes these wet environments. Going forward, I think it would be good to look in places like this, but in the Otways and maybe just over the border in South Australia, to try and figure out what the actual distribution of this species is, because with the additional collections that we made, we now have around 12 collections of this species in the herbarium. So it'd be good to figure out if it's more widely distributed or only kind of restricted to this one area. And so here's a picture of Neville with our new grass. And I'd like to say thanks to the Jim Willis donors for you know, giving young botanists a chance to have a go at taxonomy, especially grass taxonomy, and to the Botanic Gardens, and especially Neville for his great supervision. Yeah.
Thank you very much, Courtney. Great talk. I feel like it's dramatically expanded my knowledge of seed morphology terminology. There are a lot of terms in there.
Can you all hear
Looking
me?
good.
Can you all see it? Good.
Yep. All good.
Okay. Cool. All right. So, yes, before we get into it, I'd just like to acknowledge the traditional owners of the land on which I completed this project on, and which I'm presenting from now, which is the Wurundjeri people of the Kulin Nation, and pay my respect to elders past, present, and emerging. So today I'm going to be talking about Australian native truffles, and in particular, some native truffles from the family Mesophiliaceae, which is in the order Hysterangiales, which is quite a peculiar group of fungi. So what happened in this project? We've got a new genus that we've described over the course of 8 weeks that contains at least 3 new species, which I'll elaborate on further a bit later on. It's in the family Mesophiliaceae. So we looked at it, so we're characterizing specimens, we used different forms of microscopy, DNA sequence alignment, and we've also used some phylogenetic trees and written up a paper to support these new species and this new genus. So before we get into that, I just want to give you some background info on truffle-like fungi. So first main point is it's not the genus Tuva, which is the delicious truffles we all know and love and pay a lot of money for if we can afford it. This is in a different division of fungi called Ascomycetes. So it's not that. I'm going to talk a little bit about this old classification called gastroid fungi, which includes things like puffballs, earth stars, stinkhorns, and false truffles, which we're going to be looking at a bit more today. And these fungi are defined as fungi that have an internal spore mass and a loss of ballistospory. So the spores aren't ejected forcefully from the fungus to disperse them, and they're sort of just retained on the fruiting body and dispersed otherwise. We had an idea that they were polyphyletic, as in they were not related for a while, but they were finally proven like definitively in the late 90s by Hibbert and some others. So truffleization is a process that you might have heard of, which is where mushrooms or other fungi sort of squish down and become smelly and start growing underground over a very long time and to varying degrees, and they sort of become truffles. So these images on the right here, I've got different unrelated fungi that have all seemed to gradually turn into truffles to some degree. And I'd like to note that this isn't like a chronological thing. This is just a spectrum of how much mushrooms can sort of turn into truffles. And they sort of have this residual anatomy where you can see there's parts of it, especially this one that I'm waving around here, where it kind of looks like a mushroom. There's a stalk here, there's a spore mass, but it's not quite the same. And some of these features are retained as they sort of truffleize. We know that truffles most of them rely on animals for spore dispersal because they're under the ground. You know, they don't have anywhere, can't carry their spores on the wind like most mushrooms would. And we also have this group of fungi, there's sort of these ones on the right, which are cercodioid fungi, which is sort of on the way to becoming truffles, but they're not quite mushrooms, they're not quite truffles, they're somewhere in between. And I just like to include this Pedaxis pistillaris, which you might know as desert inkcap, which is sort of a classic false truffle that you see in Australia and around the world. So some more background. In the early 2000s, a Japanese mycologist, Kentaro Hosaka, published this huge paper on molecular phylogenetics of this broad group of fungi called the gomphoid phalloid fungi. And we've got this new class called Phallomystidae and 2 new orders. So this is sort of the first time that this order, Hysterangiales, which I mentioned before, sort of appeared in the literature. So it's an early diverging lineage of truffle-like fungi, which appeared around 80 million years ago, which is quite odd because when we see truffleized fungi, they usually appear at a genus or species level in like the last 60 to 20 million years. But Hysterangiales have been around as truffles for 80 million years, which is huge. And it's a whole order of fungi. So it's quite early diverging and quite a broad range of diversity that are all truffles, which is pretty peculiar. And so Hysterangiales, it's a monophyletic group. They've all come from this one ancestor, but we're not quite sure exactly what it's related to. It could be related to phallales, which you might know as stinkhorns, or gomphales, which is another order of fungi. So they're in the Phallomystidae, which is that class. But the interesting thing is, it's very obscure. We don't know what the ancestral form was. The theory's being thrown around that it might have been a stinkhorn-like ancestor or a truffle-like ancestor, and there's some version of this form, but the jury's still out on it. We really don't know what it exactly came from, and that's what makes this whole group really unique, as it's just so mysterious and untouched, I guess. So a bit more on Hysterangiales. Because they've diverged so early, they've been truffles essentially for 80 million years. They've really come a long way from what we'd consider a mushroom. So they don't have that sort of mushroomy texture. The tissue could be rubbery, gummy, or rock-like. And this sort of, goes to show how far they've diverged from other related fungi. And so this makes the ancestral form pretty difficult to interpret. And you can see it's a bit hard to sort of line up these images on the right with what you might consider to be a mushroom. You can see this one in the bottom left, there's sort of a stalk there, but again, it's a bit ambiguous. It's hard to say definitively. And they have different spore types, which I've included on the bottom left. We've got bare spores, we've got spores with a utricle, and we've got sort of ridged or ornamented spores that we see in this order of fungi. So last year, my supervisor, Naveed, who's helped me a lot on this project, he published a paper on the Hysterangiales talking about sort of some diversity in this group, because it had been recognized, in 2006 by Hosaka's paper, but we had about 7 genera in the family Mesophiliaceae at the time, which is this sort of orange family. But Naveed's paper sort of uncovered that there's a whole lot more diversity in this group. As you can see, we've got Genprov, which is a provisional genus, which is something has been identified as a new genus, but hasn't really been, described or properly looked at yet. So this is sort of just like a placeholder for what we believe is a new genus. And there's, at least 10 in here, just in this family alone. And so for this project, we're looking at this sort of provisional genus 3 and provisional genus 26. And this is sort of what we're honing in on for this project, characterizing and describing this new genus. So Mesophiliaceae, the reduced mushrooms, which I've talked about before, being false truffles, and these ones are particularly rock-like. They've been around for a while, but they're sort of restricted to Australia and Australasia, this whole family. And it's done well in an aridifying environment as Australia broke away from Antarctica about 30 million years ago. So this sort of arid tolerance wasn't really a response to Australia's changing environment, but they certainly had an edge in adapting to that change in environment. We know that they have plant and animal relationships. They form ectomycorrhizae, so they associate with eucalypts, we believe. And they also rely on animals for spore dispersal as truffles that grow under the ground. They were formerly placed in the order Lycopodales, which isn't really that related to Hysterangioles, due to their sort of powdery inner spore mass, which is a character that we see across this family, but in other groups of false truffles. They sometimes have utriculate spores, so spores with a sort of case around them, but the ones we were looking at in this project all had sort of bare spores. And they're truly subterranean. This is something also a bit interesting and unique about these truffles, is that most truffles will sort of grow just below the soil surface or a little bit in the soil surface, but these are well below the soil surface. They are at sort of the interface between where you have sort of an organic humus soil layer and sort of where that gives away to rocks and minerals. So they're, at the subsoil level almost. And this sort of helps explain why they look so much like rocks, to protect them from this harsh environment. So to characterize our specimens, we've been looking at the morphology of some herbarium specimens, and sort of measuring characters under the microscope and seeing what's different. Can we tease apart any differences between our different specimens, but also just characterize them in general, so we have something to go off, so we have a valid scientific description of them. So it took a lot of measuring, but we also had a very small sample size, which is quite an obstacle. We want a good sample size to sort of build support to identify this distinct group of fungi. So under the light microscope, we're looking at different parts of the tissue of the truffles. So the peridium, which is sort of the outer skin of the truffle, which you can see on the right here, which is very hard to sample because not only is the tissue rock-like and it's been dried out, these specimens that we were looking at are about 20 years old. It's very hard to sort of get a nice, clean section of them. And some wise words from Naveed, my supervisor, when I was struggling with this, he said, these things aren't meant to be looked at, which is really true. They grow under the ground. People haven't looked at them. They look like tiny lumps of dirt. They're just meant to be there to sort of help out with some plant-fungi relationships and provide food to animals. They're not really for human eyes to be seen and peeled apart and examined. And they are basidiomycetes, so it's been quite challenging finding basidia and characterizing them for these specimens, because as the truffles sort of age and mature, the basidia all deliquesce, so they sort of break down into nothing, and your internal spore mass really just becomes a big lump of spores, and the tissue can be very hard to sort of characterize, which is quite useful for characterizing different specimens. But it's hard to look at when it's all sort of broken down into nothing. And we've also seen some plant roots in the peridium, so the outer layer of tissue, which has been examined for related genera such as Castorium in the same family, Mesophiliaceae, by some others in the 90s. And they've been confirmed to be eucalyptus roots. Not in our specimens, but we believe so because they've been collected from underneath eucalyptus trees. And, because they've been underground for millions of years, as I've said before, it's really hard to line up the characters between the tissue, between these specimens and other more mushroom-looking fungi. So it's hard to line those up. So we also looked at them under the dissecting microscope, just to see what they look like on a macro scale. And really what we got out of this is they look like lumps of dirt. They grow in the ground and sort of, camouflaging in with dirt around them. and they're quite hard to pick up. And this sort of helps explain why we've only got, only a few specimens to look at here. If you look at this image on the left, you can see those sort of dark spots in that middle layer of the skin. They are eucalyptus roots that are sort of, facilitating nutrient exchange between the fungus and the tree. And on the sort of bottom or the left of that image, you see the locules, which is a sort of good character to look at for identifying, which is just sort of the way the spores and the structure inside in that spore mass form. So we also looked at them under the scanning electron microscope to get a good idea of the 3D surface of tissue. It's good in contrast to our light microscope, so it doesn't squish our sample up. Unfortunately, we didn't get a whole heap of useful information from our scanning electron microscope of these specimens because we'd be looking at surface texture and stuff of our spores, but being sort of bare spores and smooth, it didn't really tell us a whole lot that we didn't already see in the light microscope. So it's useful for uncovering small features. And on this image on the bottom left here, we've got some, we believe to be some eucalyptus roots that are sort of sticking out of that peridium layer. But yeah, unfortunately, it didn't add too much to our work this time, apart from some cool images. So if you're looking to, release an album of black metal music, let me know and I can probably get you some good pictures for album art for that. So to also support our new genus and species, we also, did some sequence alignment. And a lot of this was a bit of a hangover from Naveed's paper about the whole Hysterangiales. A lot of the sequences of our accessioned from that and available for us to analyze again. So we used different genes, which were ATP6, TEF1, and LSU, which are functional genes, which is really good support for divergence of these species because they're fairly conserved sequences. And we also use the ITS space region, which is good for sort of being a non-functional sort of part of sequence. It's helpful for looking at sort of fine scale divergence and differences between species. So we got good support for the genus with these sequences for the single genes on their own, and sticking them all together gave really strong support for that and for our species. The ITS data was a bit patchy. We had some missing for some specimens, though we hope to recuperate that in the future. But the big thing that happened with our sequence alignment was it included this provisional genus 26 in our new genus overall, because we're considering this to be a whole separate genus. But the sequences actually showed us that these were sort of all the one entity. They were all the same genus, which is called Fusicastorium on this phylogram in the middle here. So our new genus, we've called it Fusicastorium. This is a name that's been thrown around and attached to collections, mainly by Theresa Lavelle and Pam Catchaside and some other intrepid truffle collectors around Australia, but hadn't been formally published. It had also been, our specimen had also been called Castorium camphoratum. Castorium being in the same family, Mesophiliaceae, but not as closely related. So it's called this because it looks like Castorium and it's got fusoid spores, which just sort of elongate in shape. The whole genus, we've found that these specimens, based on notes, have a strong odor of naphthalene or camphor, or you might recognize the smell as mothballs. We also found that in the specimens that we could find Basidia for, which were mainly the couple of immature truffles that were collected, they had Basidia that had 2 spores in them, which is a useful diagnostic character. They have this 3-layered peridium, which you can see in the bottom left image. We've got an outer layer, which we call the epicutis, a middle layer called the mesocutis, which is sort of clear and contains our eucalyptus roots that are associating with the truffle, and the bottom layer called the subcutis, which is quite thin, and that's sort of like the barrier between the spore mass and the outer layer. So we know they associate with eucalypts, and they've got this sort of sandy brown spore mass overall. And so, yeah, this name, which is, been thrown around and is a useful name, just hasn't been properly described and published. So we have 3 new species for now through this study that we've recognised. We've got Fusicastorium nunguttaensis, which is from New South Wales. We've got Fusicastorium catchasidei, which was found on Kangaroo Island based off a single specimen. And we have Fusicastorium trappii, which is also from New South Wales. So I'll talk about the species a little bit more in detail. So nunguttaensis, we had 3 specimens of this one, which is in southeast New South Wales around Nungutta. And the sporocarps on these, or the truffles, have a sort of flattened shape to them. they have this uneven, lumpy surface, and the peridium's comparatively quite thick, up to about 1 millimeter in some cases. And we believe it's sister to Cachosardiae, so they have shared a recent common ancestor. So you can see the basidia on this bottom right image here, which have been stained for easier visualization. Oops, yeah. So moving on to Cachosardiae, which is our Kangaroo Island specimen. So I've only had one collection of this. There's a few sporing bodies that in this collection, but one we've been mainly looking at. And they have this quite round texture its round shape to them, and quite a thin peridium, which is a diagnostic character, really set it apart from the other two species we were looking at. And we found it was collected in a vegetation with sort of mallee eucalypts and acacia. And we can't really infer that it's particular to this sort of vegetation structure, but it was found here as opposed to more forest-like habitat. And our third species that's been described so far is Fusicastorium trappei, named after James Trappey, who's a long-time American truffle collector who's contributed a lot to this group of fungi, Hysterangiales, and collected quite a few of the specimens as well that are related and used in Naveed's earlier study. And we find it similar to Nungadaensis, but it has a sort of more even outer layer. It's not as lumpy, and a thicker mesocutis. So the middle layer of the peridium, the roots, is quite thick, up to 1 millimeter on its own, which is, set it apart from the other specimens quite a bit. And this one's sort of more genetically distinct from the other two species, and we believe that it diverged from them earlier than the other species in the genus. Yeah. And so there's more, unfortunately. So taxonomy seems to never be a complete job. We have some Western Australian specimens to look at. These images are all from Teresa Labelle, who's sort of been helping out on this project a lot, which is great. And we know that we have at least one more species just based on a cursory gene sequence analysis on these, and at least 9 more specimens to look at and characterize. And we know these ones are a bit more early diverged from the other 3 species. And Teresa's been sort of doing all the measurements and pictures for this one, which is great because the specimens are in Adelaide where she's based. So that's great of her to help out and really advance this sort of project. So why should we care about these truffles? They're everywhere. We know because they have relationships with plants and animals. They're an important food source for a lot of sort of small mycophagous marsupials, and we've found this out by analysing sort of spore content in animal scat. They're important for conservation of these animals. So in the bottom right, we've got an image of Gilbert's pottery, which is a very endangered marsupial, and it relies on truffles for a lot of its diet. And so we're hoping with a published name and description of these species, it'll be easy for people to encounter and recognize these fungi as they look like lumps of dirt. They grow under the ground. It's very hard to get many specimens of them, but we're hoping to sort of really give a clear description. It'll help people put a name to it and identify it and sort of help us contribute to our records of this. And it's going to be, that'll help us sort of build a better picture of what we recognize as this new genus Fusicastorium and sort of related truffles to this. And it's also a reference point for future studies in this group because there's a lot to be done in Mesophiliaceae, as we've sort of uncovered with the 10 provisional genera in that family. Sorry, someone trying to speak up. Anyway, so what do we want to do next? We want to characterize those Western Australian specimens. We've got more species to uncover in Fusicastorium, and there's sort of other fungi collections that have been labeled as Castorium camphoratum or Fusicastorium. So we believe there could even be more species. And from this, we're going to use it as a springboard from this study to examine infrageneric relationships within Mesophiliaceae to sort of build a better picture again of what Mesophiliaceae looks like in Australia. Furthermore, in the Mesophiliaceae family, we've got sort of the type species has sort of fallen in a weird place on the phylogram, and we need to sort of sequence that and decide, get a better picture of what Mesophiliaceae is. And for our sort of 10 other provisional genera, we're going to be sort of rinsing and repeating this sort of process, describing and characterizing our specimens and describing new species and genera, again, to sort of complete that picture of Mesophiliaceae. So yeah, that's all from me. Here's some useful papers that I really learnt on in the study. and some lovely images. And I'd just like to give some quick shouts out to all the people that sort of worked with me on this project. Naveed for supervising, Teresa for helping with our collections and descriptions, Tom, Kentaro Hosaka, and Michael Castellano, who really gave me a lot of sort of knowledge and advice on mycology, because they're all sort of veteran mycologists. It was great to hear their input. I'd like to thank the Botanical Gardens for sort of having me on board, and sort of all the other staff there for really supporting me and taking an interest in this project. And also to the Lewis Studentships and Cybeck Foundation for funding this and helping me get along with it. Yeah, that's all from me. Thanks for listening.